Population Studies, Part 2. At the end of the lesson, the student should be able to state procedures for conducting population studies, mention methods that can be used to estimate the population of organisms in a particular habitat under study, determine the population of organisms in a particular habitat under survey. Population can simply be defined as a group of organisms of the same species living in a particular habitat at a particular period of time. There are different methods by which the population of organisms can be estimated in a particular habitat, may it be aquatic, terrestrial, or arboreal. Procedures for conducting population studies include: choose the habitat to be studied, select the sampling methods to be used, collect, count, and record the different types of organisms present. Identify the different organisms. Use a key where necessary. Repeat the population studies at intervals. Collection and keeping of living organisms under study. Collected plants and parts are pressed and dried in a plant press. The name of the plant, date of collection, place of collection should be indicated. Different types of nets and traps are used in capturing and collecting animals, because animals are not stationary like plants. They move about from one place to another, and some of them move very, very fast. And animals can be collected using nets and traps from different habitats, may it be aquatic, terrestrial, or arboreal. Determining population characteristics. Population characteristics are those vital concepts that build up everything about the population. Whenever population studies is conducted, and these characteristics include the population frequency, the density of the population, and the size of the population. We are going to determine the population characteristics using the quadrat method. A list of the species in the area under survey is compiled. The quadrat is randomly tossed several times within the area. After each throw, the number of a particular species under study within the quadrat area is recorded. The recorded data is used to determine the population frequency, density, and its size. For example, studying the population of a particular grass among other grasses, and that grass name is called Tridacus protuberans. The area under study is 1,000 meters square. The total frequency of Tridacus after a total number of 100 tosses is 53, while the area of quadrat used is 1 meter square. Determination of average frequency of Tridacus. Step 1. The average frequency is determined from the total frequency of Tridacus, and it is also the total frequency of Tridacus divided by the number of tosses. The total frequency of Tridacus equals to 53 and is divided by the number of tosses, which is 100, and the answer equals to 0.53. Determination of frequency percentage of Tridacus. Step 1 prime. Frequency percentage equals to the average frequency of TriDACs multiplied by 100. The average frequency is 0.53 and is multiplied by 100, and that will give us 53%. Determination of density of TriDACs. Step 2. The density of TriDACs is the number of TriDACs per meter square of quadrat used, and the formula equals to average frequency divided by the area of quadrat used to conduct the survey. The average frequency is 0.53 and the area of quadrat used is 1 meter square, and the calculation include 0.53, that is the average frequency of the TriDACs, divided by the area of the quadrat, which is 1 meter square, and that will give us 1.53 per meter square. That is a total of 0.53 TriDACs per meter square of the quadrat used. Estimation of population size of TriDACs. Step 3. The population size of TriDACs equal to the density of TriDACs multiplied by area of habitat under study. The density of TriDACs is 0.53 per meter square multiplied by the area of habitat under study, and the area of habitat under study is 1000 meters square, and the population of TriDACs will be about 530 TriDACs. Estimation of population using the transect method. A transect is a tape marked at 1 meter interval. It is stretched across a particular area of study. The plants encountered at the interval marks are recorded. The procedure is repeated a number of times until an accurate estimate of the number and types of plants in the habitat are obtained. Can see the TriDACs and other plants within a given habitat under survey, probably a football field. The transect is stretched about 100 meters at 1 meter intervals, and a particular species of plant under study, assuming TriDACs amongst other grasses, are estimated, and the population of TriDACs can be determined within the particular field under study. The use of capture-recapture method to determine the population of organisms. In this method, animals of one type in a particular area are caught, counted, and noted as N1 and marked with ink and released. The following day, the process is repeated, and that will give you us N2, that is number of animals caught, is recorded. At the same time, the number of marked animals designated R from the previous day that we found in the sample are counted and recorded. The Lincoln Index is used to determine the population of organisms using the capture, release, recapture method. And the formula is size of population will equal to the size of first sample multiplied by the size of second sample and divided by the number of recaptures in second sample. Letter P is used to represent the size of population. Capital letter N1 is used to represent the size of first sample. And capital letter N2 is used to represent the size of second sample. While capital letter R is used to represent the number of recaptures in second sample. Worked example. Take for example, if 10 animals were trapped, marked and released. 2 days later, 20 animals were trapped. 5 of these were found to be marked. Determine the population of the animal. Solution. Population size equals to N1 times N2 divided by R. N1 represents the total number of trapped animals, and that gives us 10 animals. N2 represents the total number of animals captured, that is the second capture the following day, and that gives us a total of 20 animals. While capital letter R represents the number of marked animals among the second capture, and that gives us a total of 5 animals. We now impute in the Lincoln formula, and that will give us 10 times 20 divided by 5, and we now derive an estimate of 40 animals. Thank you for listening.